Halo, hari ini seperti biasa belajar online study with me. Hari ini aku mau belajar Bioinformatika, nyelesain course yang udah aku ambil di Coursera. Nama course-nya Bioinformatic Methods oleh Universitas Toronto. Dan kita akan mulai jam 20.40 deh. 2 menit lagi. Kita akan pakai Pomodoro 5 menit. Bentar ya. Sebentar. Sebentar. Jadi course-nya ini tuh berbayar ya sebenarnya 700.000, cuman bisa gratis kalau diterima beasiswa. Jadi kita apply dulu beasiswa dan biasa diterima sih. Jadi ini aku baru apply dan lagi nunggu, biasa 2 minggu gitu. Dan kalau di approve berarti kita bisa gratis dapat sertifikat gratis, lumayan. Dan sebelum terima financial aid-nya itu, kita udah bisa audit atau bisa akses kelasnya gitu. Dan ini aku udah sampai minggu ketiga. Rencana akan selesai di ini sih, minggu depan semoga udah selesai sampai week 8. Karena aku mau ngambil juga Bioinformatik metode 2, jadi ada 2 course, yang metode 1 sama metode 2. Dan kita bakal merekam dan berlive bersama. Oke, kita start. Kayak kena copyright gak sih? Sistem course-nya tuh ada 20 menitan lecture video sama ada lab. Di lab itu kita dikasih ini manualnya PDF, terus sambil ngerjain kuis. Kuisnya 3 soal gitu. Ini kena copyright sih, dikhawatirkan, jadi lebih bagus kita beginikan aja ya. Begini aja ya.
Welcome to the third. Lab of Bioinformatika. So what we'll be doing in this lab is we'll be doing multiple sequence alignments using a couple of different pieces of software, EGA and MAFT, in order to align protein sequences. So what we've done in the past labs is use pairwise sequence alignment methods to identify sequences that are similar to our sequence of interest from the online databases. And now in this lab, what we'll be doing is we'll be doing multiple sequence alignments to identify regions of the proteins that are similar, that align well. And this can tell us about which regions of the protein are important for biological function. So practically what we'll be doing is we'll be taking a set of sequences that are similar, and we'll be using a couple of different algorithms, so Clustall and muscle on the one hand, and MAFT on the other hand, to align parts of the protein that are similar in the related proteins. And those similar regions, those conserved regions, can give us hints about biological function. There's conservation. The assumption is that those parts of the protein are important for biological function. So multiple sequence alignments are a really powerful way to dissect proteins and to understand which parts of proteins are important for function. That's it, and I hope you enjoy the lab and lecture, and see you soon. Take care. Bye. this week's lab, we're going to be doing multiple sequence alignments. Some of the goals of multiple sequence alignments are in order to be able to perform evolutionary analyses, in order to be able to identify homology, build phylogenies, and test evolutionary models. There's also a component for multiple sequence alignments in terms of functional analysis, where we want to identify conserved regions within alignment in proteins, and also to be able to identify protein families. For structural analyses, multiple sequence alignments are useful because they can allow us to identify sequence covariation, also use them for homology modeling. And finally, there are practical applications for multiple sequence alignments, where we can use them to identify conserved primer binding sites, or to design mutagenesis experiments, also for mutant analysis. In terms of the evolutionary history, we can see in this multiple sequence alignment that the top 4 sequences all seem to have are more similar in terms of their alignment than the bottom 4 sequences, which are also aligned, in the sense that there's an insertion in the second set of 4 sequences down here. So these are likely much more closely evolutionary related than these 4 sequences, and this can tell us about the evolutionary history of the set of sequences that have been aligned. The columns in a multiple sequence alignment are indicative of homology, and the identification of homologous residues is greatly facilitated by multiple comparisons. So in this the same alignment, here I colored the residues that are conserved across all of the sequences. Either conserved based on their physicochemical properties or absolutely conserved. And you can see that if we only actually had the pairwise alignment of the last 2 sequences down here, we might think that the leucine here and the valine here are important, but in fact they aren't well conserved across all of the sequences in our alignment. Conversely, the 5th residue here, the cysteine, is absolutely conserved across all 8 sequences, and this can really tell us that that residue is very important for the function of that particular protein. So again, the identification of homologous residues is greatly facilitated by multiple comparisons. And the homology selectively is selective.
MSI identity conservers.
Be maintained due to specific structural or functional constraints. So for instance, as I mentioned in last week's lecture, the cysteines are often important for building disulfide bonds, and that is important in turn for the function of the protein. So MSAs can identify conserved or structurally equivalent residues in regions. In order to create a multiple sequence alignment, we need a system, a scoring system, and we typically use sum of pair scoring. So this is standard scoring method for MSAs. It's a column by column cost weight function, and we score using a substitution matrix, PAM or BLOSSOM typically, and we talked about those matrices last week and how they're
Sum of pairs. Scoring multiple sequence alignment. Standard MSE scoring SP scoring cost weight function.
built and what they're based on. The MSAs maximize the total alignment score by maximizing each column sum of pair scores. And the assumption here is that each column is independent of the other column. So here's an example using 3 columns in a 5 sequence multiple sequence alignment. So in column A, what we have to do is we have to do possible pairwise comparisons between sequence 1 and sequence 2 for this first. Pair, then sequence 1 and sequence 3, sequence 1 and sequence 4, sequence 1 and sequence 5, and then 2 and 3, 3 and 4, 3 and 5, and 4 and 5. And what we can do is, for each of those comparisons, we can ask, in the case of the first column, all of the residues are asparagines, they're N's, so we take the score from our Blossom matrix in this case, which is 6 for an N-N comparison, and there are 10 of those from all possible pairwise comparisons.
Kan 1, 2, 3.
So our sum of pair scores for the first column, column A, is
permutasi.
60. In column B, we don't have absolute conservation across the board. We have a mixture of N's and NC comparisons as well as CC. We could have. In the case of the NN comparisons, we've got 6 of those, and NC, we've got 4 of those. So that gives us a total column score of 24 in the case of column B. In the case of column C, we've got a mixture, again, more variability, and here our sum of pair scoring across column C is 9. So column score of 24 in the case of column B. In the case of the NN comparisons, we've got 6 of those, and NC, we've got 4 of those. So that gives us a total column score of 24 in the case of column B. In the case of column C, we've got a mixture.
Maksudnya apa?
Again, more variability, and here our sum of pair scoring across column C is 9. So there are a couple different algorithms that we'll touch on today, and we'll talk about the theory behind them. One fairly widely used method for aligning sequences is dynamic programming, and we alluded to.
Dynamic programming. Sounds familiar. Algorithm dynamic programming. Oh ya, sebenarnya ini menjeda dulu ya. Sebenarnya objektif atau tujuan dari live study with me ini, aku pengen belajar pertama adalah computer science, terutama di bidang machine learning dan deep learning selama 2023 ini, dan juga pengen belajar tentang yang lebih advance tentang kayak algoritma dan data, struktur data dan sebagainya gitu. Dan di lain sisi aku pengen mempelajari biologinya gitu, jadi terutama di genom, DNA dan sebagainya. Jadi makanya kenapa aku ngambil course Bioinformatika ini gitu. Jadi selama ke depan setiap hari Sabtu Minggu aku bakal live rencananya sih, meng up to date atau report atau apa ya bilangnya, ya istilahnya biar seru-seru aja deh. Jadi belajar dua hal. Yang pertama adalah computer science-nya, yang kedua adalah biologinya. Oke. Contohnya nih tadi nih, ini apa enggak? Nah, di sini dia ada singgung kan, algoritma yang dipakai itu dynamic programming. Nah, aku pernah baca-baca juga, dan ada yang di Coursera yang aku mau ambil tentang computer science, bahas tentang dynamic programming ini, terutama di course algoritma. Itu susah banget, tapi nanti aku ambil rencana pertengahan bulan 2023. Sekarang yang basic dulu, sampai enjoy dulu programmingnya, dulu ke yang advance.
To it last week. We could also do multidimensional dynamic programming, but this is extremely slow for large numbers of sequences. It would take 32,000 years for 10 sequences of 100 residues, so that's not so practical. So So there are progressive multiple sequence alignment methods that use profiles, and one of these that we'll talk about today is Clustall. There's also local multiple sequence alignment method called die-align, which we'll also talk about. Almost all multiple sequence alignment techniques are heuristics given the impracticality of this dynamic programming algorithm. So progressive alignment is predicated on the concept. So progressive alignment of this dynamic programming algorithm. Any two sequences that can be a algorithm. Any two sequences that can be aligned accurately can be done so using dynamic programming. Once we've made an alignment, that's equivalent to any other sequence. And that aligned set of sequences is called a profile. A pair of profiles or profiles and sequences can be aligned accurately and rapidly via dynamic programming. So it's a two sequence dynamic programming step, which is very rapid, and we can progressively align more distantly related profiles and sequences as we sort of build multiple sequence alignment. There is no separation of the alignment scoring and the alignment optimization, and there's no optimization of the global scoring function. So the concept behind profiles is the fact that two profiles can be aligned without disturbing the alignment of either individual profile. So say we have two profiles, this one over here, and this one. This is the fact that two profiles can be aligned without disturbing the alignment of either individual profile. So say we have two profiles, this one over here and this one over here. We can align those profiles without disturbing their internal alignments by either doing the following. We can join them like this. And leave this part sticking out over here, or we can add a gap here, for instance, or we can add gaps to the first profile to make to generate the alignment. And this profile alignment method is equivalent to dynamic programming with two sequences. The important thing to remember is that once we've inserted a gap, there's always a gap present. In order to for Clustall to work, we need first to compute all pairwise global alignments. And here we can either use a fast k-tuple method or a slow dynamic programming method to compute global pairwise alignments. And here we're using affine gap penalties, and this simply means that there's a sort of a, 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 scaling of the gap penalty according to the length of the gap.
Compute a previous global alignment.
And then we calculate all possible pairwise distances, and we use a distance matrix to calculate a guide tree, which is based on neighbor joining method, and it's midpoint rooted, and we'll talk about that next week. It's midpoint rooted, and we'll talk about that next week. Next we determine the order of the alignment based on the guide tree. So if our guide tree looks like this, whereby sequence 1 and sequence 2 are more closely related to each other than they are to sequence 3 and sequence 4, we would align sequence 1 and sequence 2 first, then we would align sequence 3 and sequence 4 second, then we would align the profile of sequence 1 and sequence 2 to the profile defined by the alignment of sequence 3 and sequence 4. However, if our guide tree looked like this, where sequence 1 and sequence 2 are closely related to each other, and then they're then related to sequence 3, and then those are related to sequence 4 more distantly, we would simply align sequence 1 to sequence 2 first, then we would align sequence 3 to the profile defined by the alignment of sequence 1 and sequence 2, and then we would align sequence 4 to the profile defined by sequence 1, 2, and 3, the alignment of sequence 1, 2, and 3. So really the guide tree determines the order of the alignment of the sequences to each other or the sequences to pre-existing profiles. So once we've got once we reach the step, we can perform alignments, and these, as I mentioned, can either be sequence, profile, or profile. The existing gaps are fixed.
Sequence, profile. New gaps must be inserted into all sequences.
Fixed in a profile, and new gaps must be inserted into all sequences in a profile. So here we're aligning first the most similar sequences. We've inserted a gap in sequence 2, for instance, to make the alignment better. And here's another gap inserted in sequence 3.
Insert
so that we've got these two sequences aligned to each other, the next most similar sequences, and then to align those two profiles. We would insert sequences, or we might have to insert, depending on our scoring function, some gaps here in both sequences. Sequence 1 and sequence 2.
Some gaps here in sequence 3. Gaps
or some gaps here in sequence 3 and sequence 4, and then we would use the sum of pairs scoring to calculate the overall score for the alignment. So Questable suffers from a couple of potential problems. One of them is that it's a greedy algorithm. So that means that it makes the best immediate solution, a local choice, in the hopes of finding the best overall global solution. And and that choice is made regardless of later consequences, and this means that early mistakes get propagated throughout the rest of the alignment. So, for instance, if we've got three different alignment trajectories that we could follow here. This one, or this one. You can see that we've inserted a gap in these three different locations. However, if we come up with another sequence, the later alignment actually might be best achieved with this particular solution here. However, Classwell might choose this one, so we could get stuck in a suboptimal alignment at an early alignment stage. The substitution matrices that Classwell uses are
algoritma masih belum jelas. Scoring system. Point accepted notation or PAM derived from trusted alignment between closely related sequence. Also, database uncapped multiple alignment segment. Pump numbers, evolutionary time, greater number. Okay, last number, sequence similarity. Ini ada beberapa part yang aku nggak ngerti, masih belum ngerti. Apakah kita lanjut atau mereview? Udah kayak lanjut aja dulu deh, ntar baru di review besok.
Dynamically chosen, and the distances between the sequences determines the substitution matrix, and the distances are based on the guide tree. So if the sequence identity is between 80 and 100%, a BLOSUM 80 matrix is used. If the sequence identity is between 60 and 80%, then COSTAL will use BLOSUM 60. If it's between 40 and 60%, then the BLOSUM 45 matrix is used. And if the sequence identity is below 30%, then the BLOSUM 30 matrix is chosen. COSTAL also has some tweaks that have been introduced to deal sensibly with gap penalties and with dealing with the contribution of different amino acids. Weight sequences also to reduce biases introduced by evolutionary history, so more divergent sequences will carry more weight. The gap opening penalty is actually adjusted. It's decreased for more divergent sequences.
Gap opening penalty decrease for more divergent sequence. Increase for sequence same length.
And it's increased for sequences of the same length. The extension penalty varies depending on differences in length, and there are actually position-specific gap penalties if a gap already exists at a given position, and otherwise the gap opening penalty is increased if gaps are nearby. And there are also residue-specific gap penalties. The opening penalty is decreased within a run of hydrophobic residues, which might indicate the presence of a loop structure, so we wouldn't want to disrupt that. Otherwise, we multiply the opening penalty by residue-specific values. Oftentimes tweaking the specific values. Oftentimes tweaking the parameters, such as the opening penalty, extension penalty, can influence the alignment. And what we're looking for in terms of a good alignment is to have sort of largish blocks of conservation. So here we've got like 4 large blocks of conservation with 3 insertion regions basically. And This is probably biologically more realistic in the sense that over evolutionary time, if a small piece of DNA has been acquired, it's likely to have been acquired in only one or two places, as opposed to in many different places, which would seem to be the case if we use the default parameters of cluster. So progressive alignment methods are good, fast. But as I mentioned, one of the major problems is the propagation of errors in the initial alignment through the rest of the multiple sequence alignment as it's built. And iterative methods correct for this by realigning subgroups and then realigning these subgroups into the global alignment. And the selection of groups can be...
Iterative method correct for this by repeatedly realign the subgroups and then realign this subgroup into the global alignment. Selection of group can be
based on the order of sequences on a phylogenetic tree, separation of the sequence from the rest, or random sampling. And basically what we would do here is we would do our initial pairwise alignments, the phylogenetic tree, calculate the sequence pair weights, determine the MSA, score the MSA, but then ask the question, is this alignment better than the previous round? If the answer is yes, then we go back, and if it's not, then we're done. A different kind of multiple sequence alignment is a local multiple sequence alignment, and what we're doing here is we're comparing sequence segment pairs, not the single residues. So an example of a local multiple sequence alignment program is Bioline. The segment pairs are called diagonals, and a diagonal is a gap-free pair of sequences of equal length. Mismatches are allowed. They must be consistent. I'll explain what that means in a minute. And the weights are assigned based on the alignment quality. The alignments are constructed by connecting consistent diagonals, and there's no gap penalty. Consistent diagonals. There's no gap penalty. So what are consistent sets of diagonals? So consider these two sequences, sequence 1 and sequence 2. We've got 3 diagonals here. We've got diagonal 1 and diagonal 2 here, and we've also got diagonal 3 over here. And what these diagonals mean is that this sequence region is the same as this region here. This sequence region is the same or matches up with this sequence region here, and this region matches up with this one. So the pairs of consistent diagonals are ones that don't overlap. So that don't overlap. So D1 and D3 do not overlap. D2 and D3 also do not overlap, but D1 and D2 do overlap. So the question now is which of the consistent diagonals, D1 and D3, or D2 and D3, are more significant? And this just shows that we don't want to have overlapping diagonals. So here, if we've chosen, we can see that D1 and D2 overlap. in this representation, it's quite similar to the dot matrix method of displaying an alignment that we talked about last week. Here we would have a consistent diagonals. So D1, D2, and D3 would all be consistent. This first D1 and D2 would not be consistent. So in order to choose the best sets of diagonals, we can use overlap weights, which are the weights of the diagonal and the degree of overlap with other diagonals. Basically, we use the scores for the residue pairs within a diagonal to come up with a weight of the diagonal. And then we're also interested in the length of the diagonal, how long is a region, and then we can compute a probability of finding a diagonal of that length with a score greater than or equal to this score that we calculated here when comparing two random sequences of the same length. So overlap weights favor motifs occurring on multiple sequences. And what we would do, what we end up with as the output of die-align is the set of consistent diagonals with the optimal alignment with the maximum sum of overlap weights. This is just an example with three sequences. Here's our sets of consistent diagonals as shown by these lines here, and die-align then inserts gaps where necessary. And the nice thing about die-align too is that it actually will vary and the nice thing about die-align too is that it actually will let you know which residues aren't used in the calculation of the alignment. It's especially well suited to detect local similarities in otherwise unrelated sequences, and we can use it for pairwise as well as multiple sequence alignments. And it will also align nucleotide sequences based on their amino acid alignments. So as I mentioned, die-align will indicate in its output that certain regions can't be aligned or weren't used for the alignment, and these are denoted with lowercase letters in the output. The other thing that die-align generates a standard in its output is a relative alignment score for each column in the alignment. So if you're looking along this row here, your relative alignment score, and you see high scores, that's good. So let's summarize multiple sequence alignments. What type do we use for sequences? If we've got sequences that are related over their entire length, we would probably want to do a global multiple sequence alignment as exemplified by cluster. If the sequence alignment as exemplified by cluster. If the sequences share conserved blocks separated by large insertions of unrelated material, but the blocks are generally in the same order, but may or may not be present, we probably could do a local multiple sequence alignment as exemplified by die-align. Then if the sequences contain non-alignment as exemplified by
tea coffee.
Die-align. Then if the sequences contain non-consistent sets of conserved blocks, we could do motif-based multiple sequence alignment, and this is implemented in MAFT, which we'll also be exploring today in the lab, or this week in the lab. So final hint, what about DNA versus proteins? So the thing about proteins is that they're easier to align than DNA. There's more information in proteins. We've seen in a couple of instances in these lectures that DNA basically gets a score of 1. if it matches, 0 if it doesn't. Proteins have the property of or amino acids contain the properties of being similar to other amino acids, and we can use that information to help with the alignment, and that's why we use substitution matrices. So if your DNA sequences are too divergent, to get a good alignment, you see lots of insertions or gaps being present, you can try aligning their amino acid translation and then translating the sequence back into DNA. So what we would do in this case is we would take our unaligned DNA sequences, we would translate them into protein. We've got now unaligned translated protein sequences. We would align the translated protein sequence, and then we would back translate that or reinsert the DNA information, and you would see a gap being inserted. And basically, you see gaps being inserted in triples, which is perhaps biologically more realistic than having random gaps all over the place. They just assume that if you see something that looks wrong, it probably is wrong. So wrong means lots of gaps, too many insertions, columns that don't seem to be well conserved down their length. It really does help to visualize the alignments, and what you're looking for is nice blocks of conservation with relatively few gaps inserted across the length of the sequence. All right, well, I hope you enjoyed the lab, and we'll see you next week.
Lab. Saatnya nge-lab. Saatnya nge-lab sambil nge-quiz. Sebenarnya kalau belum bayar atau belum di approve course-nya itu kita belum bisa ngisi kuis sih. Jadi kalau mau submit harus di upgrade. tapi karena nggak apa-apa, ntar kita isi aja dulu. Ntar aku ini aku jawabannya aku catat di tempat lain, jadi kalau udah di-approve ntar tinggal ngisi lagi. Pakai lab besok aja deh, kayaknya besok aja. Berhubung besok aku mau lari, jadi jangan malam-malam banget tidur. Jadi besok tinggal lab aja sih, dan itu yang lama sih. Jadi besok apa ya agendanya ya? Besok kita nyelesain week 4. Week 4 itu week 4 ujian UTS. Nah, UTS kan review NCPN 5 question. Nah, jadi besok aku kayaknya bakal lab 3, terus baca-baca lagi yang masih belum tahu. Ada beberapa yang aku catat dan belum ngerti, dan harus dicari tahu lagi supaya lebih ngerti. Dan lusa hari Selasa baru ujian UTS plus lecture week 5, yaitu tentang phylogenetics. Terus selection analysis, next generation sequencing, terus ujian akhir, terus dapat sertifikat. Minggu depan harus sudah dapat sertifikat. Jadinya kita langsung lanjut apply Bioinformatika methods 2. Ada Bioinformatika methods 2. Terus udah deh. Oke. Karena udah malam, jadi gue rencananya lanjut besok aja. Dan see you next week. Semoga next week di saat live itu kita aku udah menerima sertifikat ya, atau setidaknya ngerjain ujian akhir lah, final exam. Live minggu depan semoga final exam, dan semoga juga udah di approve financial aid-nya, jadi benar-benar bisa kita ngisi kuis. Oke.